Now we begin with cell-mediated immunity. This is part of your adaptive immunity. Just like in the innate immune system, the adaptive immune system commences with recognition of antigens. Now, pattern recognition receptors are used by cells of the innate immune system to recognize PAPs and DAMPs. But in the adaptive immune system, lymphocytes, both B and T lymphocytes, use receptors with better specificity. But B cells and T cells see or recognize antigens in different ways. So, as you can see here, on the B cell, we have the BCR on its surface. It's actually a membrane-bound immunoglobulin, and it directly recognizes antigens. Now, in contrast, in our T cells, we have TCRs, or the T cell receptor, but it cannot directly recognize specific antigens unless the antigens are bound to MHC molecules. Now, MHC molecules are present on the surfaces of the presenting cells. So the TCR does not recognize three-dimensional or unprocessed antigens. So antigens will have to be processed first and bound to this groove on the MHC before it can be recognized by TCR. These are the steps in the activation of T lymphocytes. Antigen recognition will begin the immune response. The naive T cells recognize major histocompatibility complex associated peptide antigens displayed on antigen presenting cells. The T cells respond by producing interleukin 2 and expressing interleukin 2 receptors, leading to an autocrine pathway of cell proliferation. So this is a review of cell signaling. This results to expansion of the clone of T cells that are specific for the antigen. Some of the progeny differentiate into effector cells, CD4 plus helper, or CD8 plus cytotoxic T cells. They also differentiate into memory cells. The CD4 plus helper cells activate macrophages, B cells, and other cells. The CD8 plus cytotoxic T cell kill infected target cells, and they also activate macrophages. Antigens that enter the bloodstream are captured by antigen-presenting cells in the spleen or transported to the regional lymph nodes if they are captured by dendritic cells in the skin, gastrointestinal, or respiratory tracts. So what are these APCs, or antigen-presenting cells? Now, these cells capture antigens and display them for recognition by T lymphocytes. The classical APCs are your dendritic cells, your Langerhans cells, your macrophages, and your B cells can also act as APCs. Several properties of dendritic cells actually make them the most efficient APCs for initiating primary T cell responses. So one is that they are strategically located at common sites of entry of microbes, like in the gastrointestinal tract or in the skin. And they express receptors that enable them to capture and respond to microbes. And then the activated dendritic cells migrate from the epithelia or the tissues via the lymphatics to the lymph nodes, specifically to the T cell zones. This is in response to chemokines. And then mature dendritic cells also express high levels of peptide MHC complexes, co-stimulators, and cytokines. And then we have specialized dendritic cells that are capable of cross-presenting antigens to CD8 plus T cells. Listed here are the three major activities your professional antigen presenting cells do when they encounter pathogens. So when they secrete proteins that attract and activate other immune cells, and then when they encounter the pathogens, they internalize the pathogens and digest them, and then the digested antigens are presented as peptides on their membrane surfaces via MHC class 2 molecules. And then they also upregulate co-stimulatory molecules. We will be discussing your MHC molecules and this co-stimulatory molecules in a while. So these are actually optimal for the activation of your T helper cells. Now, in the early stages of an immune response in the secondary lymphoid organs, just like your lymph nodes, your T cells encounter this professional antigen presenting cells. So mature dendritic cells are activated by microbial components via their PRRs, or their pattern recognition receptors. They express antigenic peptides in complex with high levels of MHC1 and 2 1 and 2 molecules, as you can see here. And they also express co-stimulatory ligands. So they are actually most potent activators of naive CD4 and CD8 T cells, as what we have just discussed. So they are the best activators. They are the most professional. Now, the macrophages are also found in secondary lymphoid organs, but can activate T cells also in the peripheral tissues. Just like your dendritic cells, they must be activated to become professional antigen presenting cells. They also upregulate MHC molecules and co-stimulatory ligands in response to pathogens and cytokines. These cytokines include your interferon gamma. Now, your B cells. What about your activated B cells? Now, the activated B cells interact most efficiently with differentiated T cells that are specific for the same antigen that activated them. Now, resting B cells reside in the follicles in your lymph nodes, and they gain the capacity to activate T cells after they bind the antigen through their B cell receptor. We will be discussing the details of this encounter between the T cells and the B cells later on. Now, after antigen uptake by the APCs and then antigen presentation, take note of this arrow. So it points to this co-stimulator, which we will be presenting later. Okay, so after all this, okay, the response will be having this effector T cells killing bacteria, and then you have the production and secretion of antibodies by your B cells. So the principal function for your dendritic cell is antigen presentation to naive T cells in the initiation of T cell responses to protein antigens. And then for macrophages, this is the function is antigen presentation to CD4 plus effector T cells. And this is in the effector phase of your cell-mediated immune response. And Your B cell will be doing this antigen presentation to CD4 plus helper T cells, and this is in your humoral immune response. So we will be discussing this in detail later. We have been mentioning about MHC molecules in previous slides. So what are these? We mentioned them in relation to how T cells recognize antigens. These are membrane proteins found on antigen presenting cells that display peptide antigens for recognition by T lymphocytes. So they have this peptide binding cleft to which the antigen will bind. These membrane proteins were originally identified because of their role in transplant rejection. Two classes, class 1 and 2 of MHC genes, closely linked on chromosome 6 in humans, code for HLA or human leukocyte antigens, which are the molecules critical to antigen presentation to T lymphocytes. Now, class 1 genes encode class 1 molecules expressed on the surfaces of all nucleated cells. They are expressed as two polypeptide chains. Class 2 genes encode structural glycoproteins found on dendritic cells, on macrophages, and B cells. Now this class 1, all these nucleated cells with class 1 MHC will present to CD8 plus cytotoxic T cells, while the class 2, those cells with the class 2 MHC will be presenting antigens to CD4 plus helper T cells. Let's take a look at MHC class 1 first. Antigen peptides that bind to class 1 MHC molecules are derived from viruses that have infected host cells. Degraded viral proteins are transported into the endoplasmic reticulum by specific transporter proteins. These are called your TAP, your TAP, or your transporters associated with antigen processing. The antigen peptides bind to this class 1 MHC, and then the class 1 MHC peptide complex will then be transported towards the cell surface. This can then be recognized by your CD8 plus cytotoxic T cells. Okay, so upon the transport of this complex, it will be recognized, and then the effect will be the death of the infected cell. Let's discuss MHC class 2. While viruses and some bacteria replicate in the cytosol, other bacteria replicate in cellular vesicles. In addition, pathogens can be endocytosed from the environment into endocytic vesicles. Thus, both pathogens in cellular vesicles and pathogens and antigens that come from the outside of the cell are primarily presented on MHC class 2 molecules. The class 2 molecules are present in the endocytic vesicles of macrophages, B cells, and dendritic cells. And they present antigen to CD4 plus helper T cells. So upon fusion with the endocytic vesicles, the class 2 MHC molecules become loaded with the antigen peptides. And this complex, this class 2 MHC peptide complex, is then transported to the surface where it can be recognized by CD4 T cells. Now, CD4 T cells also assist in destruction of parasites in vesicular compartments like mycobacteria by activating cells that harbor these pathogens to kill them. So, after the presentation of the antigen to the T cell, then the response is release of macrophage activating effector molecules and molecules that support B cell activation and differentiation. So what's the effect of this? The effect is killing the bacteria and parasites. And also, um, this can also support the activation of B cells. Now, this extracellular proteins that will be presented to the Th2 type of helper T cells will respond by activating B cells, and you also have secretion of of antibodies to eliminate bacteria and toxins. So for extracellular parasites, your CD4 T cells can activate your macrophages to endocytose and destroy pathogens, as well as instruct B cells to produce antibody to opsonize the pathogen. So two types of or two subsets of CD4 plus cells are involved here. You have your Th1 and you have your Th2 subsets. We see here the interaction between antigen presenting cell, which is on the top portion, and the alpha beta T lymphocyte at the bottom. Some experts refer to this set of protein to protein interaction as immunologic synapse. So the schematic structure of a class 1 MHC and class 2 MHC proteins can be seen here, bound to antigen fragment here in blue. So the antigen binding pocket is at the top. So this is inverted. So it is at the top of the MHC molecules here, and they are formed by the alpha 1 and alpha 2 parts of the molecule in your class 1, and alpha 1 and beta 1 of your class 2 MHC molecules. So you can see the difference in the structure of these two classes of MHCs. So you can also see the structure of your TCRs, or your T cell receptors. So your helper, your CD4 plus helper T cells recognize peptide antigens in the context of MHC class 2 molecules that are expressed in dendritic cells, macrophages, and B cells, while your CD8 plus cytotoxic T cells recognize peptides associated with this class 1 MHC molecules. What happens after antigen recognition? Let's take a look at this dendritic cell. This immature dendritic cell, using its TLR, or the toll-like receptors, will be able to recognize the bacterium in a tissue. It processes the bacterial proteins and then upregulates the expression of MHC class 2 and co-stimulator molecule B7. We'll be looking at this in a while. It also changes expression of chemokine receptors, and as this changes in the dendritic cell occurs, the dendritic cell becomes mature. It then migrates towards the T cell area of the lymph node that drains this tissue, and it goes through the lymph node through this afferent lymphatics. It then migrates to the T cell area here, okay, and presents the peptides to the CD4 plus T cell with the appropriate TCR. We talked about T cell and APC interaction a while ago, so let's talk about it in more detail. So three signals are required for this interaction to be successful. Signal 1 is provided by the TCR MHC peptide interaction along with CD4 and CD8 co-receptors here. Signal 2 is provided by the engagement of co-stimulatory molecules. It's a separate set of molecules, and the best characterized co-stimulatory molecule is B7. And it is shown here. B7 is present in many APCs, and it binds to the CD28 present on the T cell. So this is your signal 2, your co-stimulation. Now, signal 1 and 2 will initiate signal transduction cascade that results in activation of transcription factors and cytokines, which now provides signal 3. Now, the cytokines can act in an autocrine manner. So, this interleukin 2 can act on this T cell, and then it can also act in a paracrine manner. So you have this interleukin 2 that will stimulate neighboring cells. After this, there will be direct T cell proliferation and differentiation. Now, if the T cell receives signal 1 and not signal 2, then the T cell is turned off. Let's see again the sequence of events in T cell responses. So after antigen recognition by T cells, it induces the production of cytokine, particularly in CD4 plus cells. This leads to clonal expansion as a result of cell proliferation, and differentiation of T cells into effector cells or memory cells happen. Now, in the effector phase of the response, effector CD4 plus T cells respond to antigen by producing cytokines that have several actions, such as recruitment and activation of leukocytes and activation of B cells, while CD8 plus cytotoxic T lymphocytes respond by killing other cells, specifically those cells that are infected. They also activate macrophages. Memory T cells are long-lived and have enhanced ability to react against antigens. Now, the effector T cells recognize antigens in the lymphoid organs or in peripheral non-lymphoid tissues. Now, these cell responses should decline after the antigen is eliminated. We look at one of the effector cells of cell-mediated immunity, and this is your cytotoxic T cell. Okay, so this photo shows us two cytotoxic T lymphocytes that bind to this tumor-specific antigen on the surface of a cancer cell. So they are delivering the kiss of death to induce apoptosis. How will the cytotoxic T cell kill its target cell? The targets, of course, are those cells that express MHC class 1 peptide complexes. Apoptosis of the target is induced by two ways. First is the action of the granule contents of the activated cytotoxic T cell, and the other one is through this fast ligand fast interaction. The predominant pathway involves the action of granule contents within the activated CD8 plus T cell. The cytotoxic T cell releases the granules onto the target cell by exocytosis. A key constituent of the granules is your perforin. This is similar to the complement membrane, to that of the complement MAC, which we described. And this forms pores in the target cell membrane. Now, through these pores, serine proteases, these granzymes, pass into the target cell to induce cell death. And then a small protein, not shown here, also targets the enters the target cell, and it activates pathways that also lead to the killing of this time killing of intracellular bacteria which live inside macrophages or dendritic cells. A second pathway occurs via interaction of the molecule FAS-L expressed on cytotoxic T cells with FAS that are expressed on many host cells. This interaction causes sequential activation of caspases. This is a set of proteolytic enzymes found inside target cells. And this causes cell death within hours. Since death by apoptosis does not result in the release of the contents of the cells, it will not stimulate an inflammatory response. And this may prevent the spread of infectious virus into other cells. Let us take a look at the helper T cell. A naive CD4 plus T cell may differentiate into subsets that produce different cytokines that recruit and activate different cell types. So, how will the CD4 plus T cell do this? Okay, so when the PRRs on APCs interact with these PUPs from pathogens, this will determine which polarizing cytokines are produced and into which T helper cell a naive T cell will differentiate. These polarizing cytokines that arise from dendritic cells or other neighboring cells interact with the cytokine receptors present on the T cell, and this generates signals so that there is a transcription of this master gene regulators. These master gene regulators in turn regulate gene expression, including effector cytokines, which define the function of each subset. These are the T helper subsets we were referring to in the previous slide. The figure seems intimidating, but it just synthesizes the distinguishing characteristics of this T helper cell, their differentiation and activity. The polarizing cytokines, the master transcriptional regulator, the effector cytokines, and the effector functions are depicted for each of the major helper subsets. You do not need to memorize this, but please remember the principal target cells for, especially for TH1, which is your macrophage, and then for TH2, the principal target cell is your eosinophils, since it combats helminth infections and activates eosinophils. And then you also have your neutrophils as the target for TH17. For this TFH cell, FH stands for follicular helper. Okay, so this is your follicular helper T cell. Okay, so these are found in the follicles in the lymph nodes. So the target cells for this are your B cells. So they regulate the affinity maturation of the B cells in the germinal center. We will be looking at that later. Another subset or subtype is the Treg. This is your regulatory T cell, and it's very important because it suppresses the immune response. I just said in the previous slide that regulatory T cells are important for suppressing immune responses. So the next question is, how does it regulate immune responses? So this diagram shows some possible mechanisms for the activity of these regulatory T cells. First, we have cytokine deprivation. These T regulatory cells express high levels of high affinity interleukin 2 receptors, and they can compete for the cytokines that are needed to activate cells. Second, we have cytokine inhibition. The Tregs can secrete several cytokines, including interleukin 10 and the TGF beta, or your transforming growth factor beta, which bind receptors on activated T cells and therefore reduce signaling activity. Third, we have inhibition of antigen presenting cells. So the Tregs can interact directly with MHC class 2 expressing antigen presenting cells and inhibit their maturation. Therefore, they cannot present antigens. And then number 4, we have cytotoxicity. Tregs can display cytotoxic function and kill cells by secreting perforins and granzyme. We mentioned about co-stimulatory molecules a while back. If we have co-stimulatory molecules, we also have co-inhibitory molecules. These molecules antagonize T cell activating signals. Co-inhibitory receptors are present in both T and B cells. They are also present on NK cells, dendritic cells, and macrophages. Now, co-inhibitory receptors are not present in naive cells because naive cells should be allowed to mature, to proliferate and differentiate. Upregulation of these co-inhibitory signals happen towards the end of the immune response when proliferation of cells is no longer needed. Now, in the research for cancer immunotherapy, they are looking into checkpoint inhibitors because this may be used to activate this T and B cells. Now, what is the fate of your T helper cells? One of the fates of your T helper cells is that it helps in B cell production of antibodies, and it also helps in the production of B cell memory cells. So this slide actually acts as a segue for the next type of adaptive immunity, which is your humoral immunity.